Let's start with today's featured paper, combining machine learning and iterative experiments to keep pace with emerging viral variants of concern. By Thomas Sheffield and Lou Tan Mai Pham from Biosecurity and Bioassurance, Sandia National Laboratories, Livermore, California, United States of America. This paper was selected because it introduces a highly novel and scalable framework that integrates machine learning with iterative experiments to proactively predict emerging viral variants, which is crucial for global pandemic preparedness and rapid adaptation of medical countermeasures. Here's a full breakdown. Hello, everyone. Today, we're diving into a crucial challenge in pandemic preparedness. How do we stay ahead of rapidly evolving viruses? The core question this research tackles is how to effectively predict and evaluate new viral variants of concern. It's vital because current methods often fall behind as viruses mutate, making it hard to update our defenses like vaccines and diagnostics in time. The researchers developed a sophisticated framework using SARS-CoV-2 variants as their model system. Their approach ingeniously combined machine learning models, specifically random forest and neural networks, with targeted high-throughput experiments. They measured critical factors like binding to human ACE2, RBD expression, and antibody escape. This wasn't a one-off study. It involved an iterative refinement process, continuously integrating new experimental data from over 200 variants derived from wild-type and Omicron strains. The results were quite impressive. The machine learning models achieved a high accuracy, with correlation coefficients of up to 0.79 for antibody binding. This predictive power was further enhanced by iteratively integrating experimental data from over 200 variants. Importantly, the models demonstrated strong generalizability across diverse antibody types, including heavy chain-only antibodies, by effectively encoding specific input features. This means the framework successfully learned to predict complex viral behaviors. So what does this mean for us? This dynamic framework offers a powerful tool for the rapid assessment of emerging viral variants, which is critical for guiding therapeutic strategies and vaccine updates. While it does rely on iterative experimental data for continuous refinement, this investment is key to maintaining predictive power. Ultimately, this research paves the way for a more proactive and data-driven response to future viral threats, significantly bolstering our pandemic preparedness efforts. That was our featured study. Today, PubMed indexed 2,957 new open access papers across 638 journals. We've selected 10 trending research areas and 10 studies for today's video. Let's look at the trending topics before we continue. Today, the research landscape is buzzing with advancements in clinical health and patient care, alongside fascinating new developments in environmental biology and materials science. Now let's see which papers stood out. We've finally cracked direct mitochondrial DNA editing, a groundbreaking step for severe diseases. Plus, a simple urine test could revolutionize global HPV screening and cervical cancer prevention. Now let's explore each of today's trends in detail, starting with number 1. Trend 1 of 10, environmental and plant biology. This trend explores plant biology, including genetics, nutrition, and ecological interactions, alongside broader environmental studies such as pollution and traditional knowledge. This cluster covers 206 papers. Representative work includes bryophytes of Benin Republic, traditional knowledge, uses and perceived threats, species-specific performance, and nutritional responses of radish and red beet to zinc manganese in sand culture, and atmospheric microplastic deposition in high-altitude snowpacks across Turkey. Trend 2 of 10, clinical diagnostics and public health. This trend examines diagnostic methods, their clinical utility, and public health implications for disease detection, management, and healthcare optimization. This cluster covers 292 papers. Representative work includes antiviral resistance in influenza, clinical and public health implications, predictive variables for early detection of sepsis in adults admitted to intensive care units, protocol for a systematic review, and evaluating a quality improvement learning collaborative, qualitative evaluation of using labs wisely. Trend 3 of 10, neuroscience and brain research. This trend explores diverse aspects of brain function, neurological disorders, and advanced research techniques in neuroscience. This cluster covers 196 papers. Representative work includes population-based brain templates for ultra-low field MRI, hippocampal neuroinflammation, and altered peripheral neurobiological protein profile in experimental arthritis and systemic juvenile idiopathic arthritis, and predictive networks generate motion-induced color illusions. Trend 4 of 10, healthcare management and patient experience. This trend explores strategies for healthcare delivery, patient well-being, and the dissemination of medical knowledge. This cluster covers 419 papers. Representative work includes implementation strategies for evidence-based management of type 2 diabetes at primary healthcare facilities in low- and middle-income countries, and related barriers and facilitators. A scoping review, beyond survival, reconstructing body image and femininity after breast cancer, and a bibliometric assessment of the Turkish Journal of Orthodontics after WAS indexing. Trend 5 of 10, surgical procedures and medical interventions. Researching various medical and surgical procedures, their techniques, outcomes, training, and related interventions. This cluster covers 376 papers. Representative work includes impact of opioid-free anesthesia on postoperative nausea and vomiting, chronic pain, and quality of recovery in patients undergoing video-assisted thoracoscopic surgery. A systematic review and meta-analysis of randomized controlled trials, training in pleural interventions, not just a matter of rights, and right anterolateral mini-thoracotomy, and full sternotomy approaches for wheat procedure, a propensity-matched comparison. Trend 6 of 10, animal and veterinary science. Research exploring animal health, physiology, disease, and production across various species. This cluster covers 159 papers. Representative work includes heat stress response of immortalized cumulus cells derived from Hanwu cattle, proliferative and granulomatous ileitis in a pig naturally infected with Lawsonia intracellularis, and effects of chicken liver hydrolysate paste addition on structure-function relationships in emulsion-type sausages. Trend 7 of 10, pharmacology, toxicology, and disease mechanisms. Studies drug actions, toxic effects, and disease mechanisms to combat pathogens, mitigate injury, and understand biological processes. This cluster covers 321 papers. Representative work includes the Trojan horse against antimicrobial resistance, siderophore conjugates, and public health. Comparative study on the protective effects of Silybum marinamel, seeds, and leaves against nickel chloride-induced anemia, liver and kidney injury. An in vivo and in silico approach, an inhibitory effect of acetylchaconin on drug-resistant Candida albicans. Trend 8 of 10, public health and lifestyle epidemiology. This trend examines how lifestyle factors, environmental exposures, and public health interventions influence population health outcomes using epidemiological methods. This cluster covers 270 papers. Representative work includes trends and determinants of tobacco use among women of reproductive age in East Africa, a trend in multilevel analysis using demographic and health survey data, emerging evidence for skeletal muscle as a target of BPA exposure, and reduction of salt in the diet and improvement in blood pressure, low cost, robust evidence, and challenges in implementation. Trend 9 of 10, materials science and applied physics. Investigating material properties, development, and advanced physical applications across diverse scales and systems. This cluster covers 316 papers. Representative work includes effective charge saturation in ferritin cages, mechanical properties of concrete reinforced with Elias guineensis midrib fibers, and atomistic insights into the magnetic field modulation of the formula. See text, adenosine receptor. Trend 10 of 10, cancer diagnostics and therapy. This trend explores advanced methods and strategies for cancer detection, imaging, and therapeutic interventions. This cluster covers 402 papers. Representative work includes first-in-human evaluation of 18F, alpha-fenotensin for NTSR1, targeted imaging of prostate cancer, a head-to-head comparison with 68, GA, PSMA, 617, risk-adapted SBRT, formula. See text. For recurrent lung-only metastases in patients with frailty or ultracentral anatomy, and TRF and gastric cancer. Molecular mechanism exploration and novel strategies for precision diagnosis and therapy. That's the full research landscape for today. Remember, our number 1 paper, Combining Machine Learning and Iterative Experiments 2, was already covered at the top. Now let's continue with papers 2 through 10, one by one. Up next, our second paper out of 10, Development of a Novel, Urine-based high-risk human papillomavirus polymerase chain reaction test to predict cervical intraepithelial neoplasia abnormalities associated with cervical cancer by Vasu Sani and Ricky Tichiu from Phase Scientific International Limited, Hong Kong. This paper was selected because it introduces a novel, non-invasive urine-based test for high-risk HPV, offering a significant advancement in cervical cancer screening with immense potential for global public health impact, particularly in improving accessibility and uptake. Let's break down its key contributions. Cervical cancer is a serious health issue, and effective screening is vital for early detection and treatment. However, traditional screening methods can be invasive, leading to lower participation rates. This study aimed to address this challenge by developing a novel, non-invasive urine-based test for high-risk HPV, hoping to make screening more accessible and improve early detection of precancerous lesions. Researchers designed this study in 2 cohorts. The first cohort included 82 women with confirmed SIN2+ lesions, where they compared the new urine test against standard cervical brush samples. The second, larger cohort of 983 women was used to optimize the test's performance, specifically adjusting the cycle threshold cutoffs to achieve the best balance of sensitivity and specificity for detecting precancerous lesions. Up next, our third paper out of 10, comprehensive assessment of on- and off-target mutagenesis via lipid nanoparticle delivery of CRISPR-Cas9 genome editing by Yuichi Nao and Akitsuhata from Department of Clinical Application, Center for iPSC Research and Application, CIRA, Kyoto University, Kyoto, Japan. This paper was selected because it provides a comprehensive and experimentally validated assessment of on- and off-target mutagenesis for CRISPR-Cas9 delivered via lipid nanoparticles, directly addressing a critical safety concern that is paramount for the clinical translation and widespread therapeutic application of this transformative gene editing technology. Let's break down its key contributions. CRISPR-Cas9 genome editing holds immense promise, but its safe application, especially in therapies, hinges on understanding its potential for unintended genetic changes. Specifically, when CRISPR is delivered using lipid nanoparticles, or LNPs, we need to know if it's truly safer than other methods. This study aimed to fill that knowledge gap by rigorously evaluating both on-target and off-target effects. To tackle this, researchers used a multi-pronged approach. They studied mouse muscle and human iPSCs, employing a range of advanced techniques. This included amplicon sequencing for on-target effects, benchmarking in silico prediction tools, and developing a novel indel cluster method with whole genome sequencing to detect off-target mutations. Up next, our fourth paper out of 10, Antagonizing NRG1-ERBB4 signaling pathway with spironolactone for the treatment of schizophrenia. Results of a randomized controlled drug repositioning clinical trial by Alkamayat Hassan and Peter Fokai from Department of Psychiatry, Psychotherapy and Psychosomatics, Medical Faculty, University of Augsburg, Germany. This paper was selected because it presents the results of a randomized controlled clinical trial, a critical step in drug development, demonstrating a novel drug repositioning strategy for schizophrenia by targeting the NRG1-ERBB4 signaling pathway, which addresses a major unmet medical need. Let's break down its key contributions. Today, we're looking at a study exploring a new approach to treating cognitive issues in schizophrenia. The NRG1-ERBB4 signaling pathway is known to play a role in these cognitive impairments. Interestingly, an existing drug called spironolactone has shown promise in modulating this pathway in lab studies, leading researchers to investigate its potential in patients. This was a multi-site, proof-of-concept randomized controlled trial. Researchers enrolled 90 patients with schizophrenia and randomly assigned them to receive either spironolactone at 2 different dosages or a placebo for 3 weeks. The main goal was to see how these treatments affected working memory performance, a key cognitive function. Up next, our fifth paper out of 10, Lipids and Lipid Nanoparticles Functionalized with Randomized Polyethylene Glycol, RPG, for mRNA delivery by Philip Dreyer and Holger Frey from Department of Chemistry, Johannes Gutenberg University Mainz Duisburgweg, 10-14 Mainz, Germany. This paper was selected because it introduces a novel and critical solution to the growing problem of anti-PG antibodies, which significantly limits the efficacy and safety of mRNA therapeutics, thereby advancing the state-of-the-art in a revolutionary drug delivery platform. Let's break down its key contributions. PEG-related lipid nanoparticles are essential for delivering therapeutic mRNA, but they face a significant challenge. The immune system can develop anti-PG antibodies. These antibodies recognize and clear the nanoparticles too quickly, diminishing their effectiveness. This study aimed to address this problem by exploring a new approach to maintain the benefits of PEG-ylation while avoiding this immune response. Researchers synthesized randomized PEG, or RPG, using a technique called living anionic ring opening copolymerization. They then functionalized these RPEGs with lipid anchor groups to create RPG lipids. To test their hypothesis, They investigated anti-PEG antibody interaction using ELISA and X-ray reflectometry and formulated LNPs with these new rPEG lipids. They compared these novel LNPs to established PEG-based LNPs across various performance metrics. Up next, our 6th paper out of 10. Guide RNA reprogramming facilitates minimized tracrRNA-dependent off-target and versatile CRISPR/Cas9 engineering by Wenxia Yu and Yunbo Xiao from Department of Plastic and Reconstructive Surgery, 9th People's Hospital, Shanghai Jiao Tong University School of Medicine, Shanghai Institute of Precision Medicine, Shanghai, China. This paper was selected because it introduces a novel guide RNA reprogramming strategy, informed by machine learning, that significantly minimizes tracrRNA-dependent off-target effects, thereby enhancing the safety and versatility of CRISPR/Cas9 systems for broad biomedical applications. Let's break down its key contributions. CRISPR-Cas9 technology has revolutionized genetic engineering, but its widespread application is hindered by safety concerns, particularly unintended off-target edits. This research delves into a previously overlooked mechanism of off-targeting, aiming to make this powerful tool safer and more versatile. It's a critical step towards realizing the full potential of gene editing. Researchers employed a sophisticated approach, utilizing machine learning trained on high-throughput screens to understand gRNA behavior. They investigated the crRNA, tracrRNA duplex in human and mouse systems, leading to the discovery of a new off-target mechanism. This detailed analysis allowed them to engineer improved CRISPR components for enhanced safety and utility. Up next, our 7th paper out of 10. Mitochondrial genome microhomology-mediated editing by donor DNA delivery into mitochondria in human cells by Vadim V. Maximov and Nina Antelis from Pfizer Ltd., Jerusalem 972-6313, Israel. This paper was selected because it introduces a groundbreaking and highly novel strategy for direct correction of mitochondrial DNA mutations, addressing a critical unmet need for severe human diseases that currently lack efficient therapies. Let's break down its key contributions. Many severe human diseases are caused by mutations in our mitochondrial DNA, and unfortunately, effective treatments are currently lacking. This presents a significant challenge in biomedical research. Our study aimed to explore a novel strategy for directly correcting these mtDNA mutations, which could potentially offer a much-needed cure for these devastating conditions. We investigated this by using cultured human cells and human mitochondrial lysates as our model systems. Our primary technique involved delivering double-stranded DNA oligonucleotides directly into mitochondria, leveraging the cell's own microhomology-mediated end-joining pathway. We also explored combining this with the CRISPR-MITO AsCas12a system to enhance editing efficiency. Up next, our 8th paper out of 10. Alert on a novel South American bat coronavirus with a Furin-Cleavage site. A One Health early warning by Lucas Alves de Moda Santana and Lisandro Pinto Borges from Universidade Federal de Sergipe, Departamento de Odontologia, Aracaju, SE, Brazil. This paper was selected because it provides a critical One Health early warning about the discovery of a novel bat coronavirus possessing a Furin-Cleavage site, directly addressing the most pressing global challenge of pandemic preparedness and zoonotic spillover. Let's break down its key contributions. Good morning, everyone. Today, we're discussing a critical alert regarding a newly identified coronavirus. Coronaviruses, as we've learned, pose significant global health challenges, and understanding their origins and characteristics is paramount. This paper highlights a novel virus found in bats, which are known natural reservoirs for many coronaviruses, including those with pandemic potential. The researchers conducted a basic science study focusing on viral surveillance in South American bat populations. They utilized advanced genomic sequencing and bioinformatics tools to characterize any novel viruses. This approach allowed them to identify and analyze the genetic makeup of the newly discovered coronavirus, specifically looking for features that might indicate its potential threat. Up next, our 9th paper out of 10. Proteomic health archetypes identified in disease-free adults enable risk assessment for diverse chronic diseases by Xiangyang Zhang and Jiang Zhu from National Clinical Research Center for Geriatric Disease, Xianya Hospital, Central South University, Changsha, Hunan, China. This paper was selected because it introduces a novel proteomic framework to define proteohealth archetypes in disease-free adults, enabling a systems-level, multi-disease risk assessment that significantly advances the state-of-the-art in personalized preventive medicine. Let's break down its key contributions. Preventing chronic diseases is a major challenge, partly because we lack good ways to identify people at high risk. The plasma proteome, which is the complete set of proteins in our blood, holds great promise for a comprehensive health assessment. However, its clinical use has been limited by technical biases and a tendency to focus on one disease at a time. This study aimed to overcome these hurdles. Researchers studied a large group of nearly 12,000 disease-free adults to define 5 distinct proteohealth archetypes, or PHAs, using advanced unsupervised learning. They developed a robust classifier based on protein ratios, ensuring it could work across different measurement platforms. This innovative design allowed them to assess how these archetypes predicted the risk of various chronic diseases and even survival over time. Up next, our 10th paper out of 10, BCL6 in T cells promotes type 1 diabetes by redirecting fates of insulin-autoreactive B lymphocytes by Landon M. Clark and Rachel H. Bonami from Department of Medicine, Division of Rheumatology and Immunology, Vanderbilt University Medical Center, Nashville, Tennessee, USA. This paper was selected because it identifies a novel and selective therapeutic target, BCL6 in T cells, which fundamentally advances the understanding of type 1 diabetes pathogenesis and offers a promising pathway for highly specific interventions beyond broad immunosuppression. Let's break down its key contributions. Type 1 diabetes is a challenging autoimmune disease where the body's immune system attacks insulin-producing cells. Current treatments often broadly target T cells, but they only delay the disease, not fully prevent it. This really underscores the urgent need for more precise and effective therapeutic strategies. Researchers investigated this by using an experimental model of type 1 diabetes. They specifically manipulated BCL6 expression in T cells, using techniques like adaptive transfer to observe its impact. The core of their approach was to understand how T-cell BCL6 influences the behavior of insulin-targeting B cells within the pancreas and lymph nodes. That's all for today's PubMed Daily. If you found this helpful, please like, leave a comment, and subscribe for daily paper summaries. See you tomorrow.